It is 1 o'clock. So we're going to go ahead and jump in today. Welcome to the first of our webinar series. Hopefully you're seeing my screen now. Bottom up focus on forage. I'm Rebecca Kern. I'll be the host for most of this series. We're going to go from the alfalfa breeding and understanding the genetics and quality traits. We have David Mickelson here with us today from S&W Seed Company. Then on March 15th, we're going to learn about selecting seed for our operation and our goals. We're going to get some producer insights in April, and then we will talk about laboratory testing in May. So just some housekeeping things for you all. I'm going to be monitoring the chat and the Q&A as David goes through his presentation. So go ahead and put your questions, concerns in those places. We also have gotten some emails already with some questions. So I know we're going to have a great discussion afterwards. So without any further interruption, I think I'll go ahead and introduce my colleague that I'm very excited to host here from the consortium, Dave Mickelson. So David Mickelson has been an alfalfa breeder with S&W Seed Company in Arlington, Wisconsin since 2016, working on developing dormant and non-dormant varieties for worldwide use. Prior to that, he was in alfalfa research with Pioneer Hybrid for 24 years, running yield trials, testing, and selecting for pest resistance and quality to produce dormant, conventional, leafhopper resistant, Roundup ready, and reduced lignin varieties for North America. He holds a Bachelor's of Science in Agronomy and Agricultural Economics from the University of Wisconsin. He is the past president and member of Industry Extension Forage Advisory Committee and member of Midwest Forage Association and the American Forage and Grasslands Council. He serves on the board of the Near-Infrared Spectroscopy Forage and Feed Testing Consortium, promoting the use of NIRS for forage and feed analysis. He is currently vice president of his county farm bureau, president of his church, and leader and supporter of local 4-H and FFA. He lives near Arlington, Wisconsin with his wife and 2 children, where he is still part of his 300-acre family farm he grew up on, raising corn, soybeans, wheat, forage, and he helps his son show Hereford cattle and crossbred sheep. In the winter, he also curls at his local curling club. So without any further, I'll allow you to go ahead and share your screen, Dave, and everyone else, I'll be monitoring the chat and looking for the comments.
All right, well, thank you very much, Becca, and I hope everyone can see the screen with my picture on it now. My name is Dave Mickelson, and I'm with S&W Seeds, and hopefully, come on, some of you have heard of S&W Seed Company before. S&W is a seed company started by 2 former farmers in California named Silvera and Williams, where the S&W comes from. And in the 1980s, they started breeding their own alfalfa varieties for salt tolerance that they could grow on their farm there. And these were non-dormant varieties.
Hey, Dave, not to interrupt you, but I don't think you're sharing your screen.
Oh, hold on, there we go. Because I didn't hit the share button. You know, I've done this not that many times. So now we can see it, right?
Yes, looks great.
Okay,
thanks, Dave.
Okay, there we go. So these 2 farmers started breeding their own alfalfa in non-dormant, and as they sold alfalfa to other farmers, it kind of took off. Over the years, they sold more and more. They acquired some companies, including one in Australia, and it started to go worldwide to where they became the world's leading non-dormant alfalfa variety company. And so that led to this vision that they had to be the world's preferred proprietary seed company. So today, S&W is involved not only just in alfalfa, but also forage and grain sorghum, also sunflower breeding, and we also have a little bit of the natural sweetener stevia. So when I started, I was with Pioneer And then at the beginning of 2016, S&W acquired the alfalfa breeding program, the R&D, and also the alfalfa production facilities from, at the time, DuPont Pioneer. And so it kind of brought together the dormant program from Pioneer and the non-dormant from S&W, and it really worked out as a nice fit for an alfalfa seed company. So back when I started, there was probably in the 90s, there were over 20 individual companies all breeding alfalfa. There's been a lot of consolidation over the last 20, 25 years, where right now we're down to 4 current private breeding programs that are providing new genetics to the alfalfa industry. So no matter what alfalfa variety you're planting, whatever brand, if you've bought it in the Midwest, it's going to come from one of these 4 companies from our breeding programs, from S&W, which came out of the DuPont Pioneer program. There's Forage Genetics International, owned by Land O'Lakes. And over the years, they've acquired a number of other brands, ABI, ABT, the WL brand, and others. We have Alfalfa, which was the Cal West breeding program, also out of Wisconsin. And then they merged with the Dairyland program. They were acquired by Dow Mickelson. And then when Dow merged with DuPont and became Corteva, that's how Alfalfa ended up there. Legacy Seeds also has their own breeding program now. They're also based in Wisconsin, and they actually just built a new research and greenhouse facility here in Wisconsin. At the same time, over the last 25 years, there's been a decrease in funding for public breeding. And so our universities now, they do they used to release their own varieties, but now they do work on primarily base germplasm improvement for others. And they also do a lot of the basic science research and the physiology and the agronomic research that the breeding companies really don't have time to do anymore. So no matter what company you're with, it's, we're all trying to produce the best possible product for the farmer and then ultimately for the animal that uses that product. And we're trying to do that through, leading research and development. So today I'm going to talk about some of those processes, either a couple of classical breeding strategies that we use, and then later talk about some of the newer biotech things that we're able to do. As we start here, I want to just talk a minute about the near-infrared spectroscopy, the Forage and Feed Testing Consortium. And what that is together we're an association of commercial labs, universities, government groups, plant research entities like us, and then also the 4 major NIR instrument companies. And we've gotten together to collaborate, and together our mission is to promote and standardize the use of the NIRS through the development of robust, accurate prediction calibrations. And what this does is it helps to reduce the variation between instruments and between labs, so we all have greater precision and accuracy and make things better for all of us. So, on the lab side, Ward Labs is a good partner in the consortium. There's many other labs that take advantage of this, not all of them, but a lot of them. We have the USDA labs involved, state and university labs. And at S&W, we're one of 3 out of the 4 breeders that use the consortium equations when doing our quality analysis. So we use that to develop new genetic products. So the NIRS, it's a good tool for us to quickly analyze forages for different quality constituents. And part of the power of that comes from developing these equations with samples from all over the United States. A lot of different varieties, geographies, environments. They just make the calibration better and really helps all of us. So as researchers, we use the NRS because it's very cost effective versus wet chemistry. Wet chemistry is sort of the gold standard. But what we need is we need rapid turnaround time. And I know everybody wants that. But in breeding, there's times when we need results to be done and processed in a timely manner so that we have time to use it and get the plants that we need. If we don't get the results in time, it's really, it's not useful to us. And by using NIR, so for instance, S&W, we do enough samples, thousands of samples, that we can have our own machine. And we can analyze for many different constituents we don't have to worry about the cost as much as if we were sending it to a lab and trying to limit that. So when you're thinking about picking an alfalfa variety, you're probably asking yourself a number of questions. So what's important to you? You have to know the soil that you're going on. You know your farm. Maybe you like certain seed treatments or want that. Do you have lodging issues? Do you have weed issues when you're trying to establish alfalfa? If you had to choose between quality and yield, which would you choose? I know everybody says, well, I want both, but if you had to choose one, which would it be? And do you know what pests you typically would see on your farm? So those are all questions you're asking yourself. And when we talk about alfalfa genetics, I like to tell people that they're a little more complex than some of the other crops. But it's not like corn where you have inbred parents and you're making a true hybrid to get that hybrid vigor. It's not like wheat or soybeans where we have a self-pollinated crop or a pure line. In those cases, you buy a bag of seed. Every seed in the bag is supposed to be the same or have the same genetics so that it's very uniform. With alfalfa, we characterize populations in that bag. It's a population of seeds characterized by different percentages so that every seed in the bag is actually genetically different. So one way to think about that is, different alfalfa seeds in a bag. You can kind of think about that like all the people that are on this call or on this webinar. We're all different, yet we have some things in common. So we all are interested in forages, but we can characterize ourselves in percentages by saying, okay, a certain number of us are right-handed and a certain percentage are left-handed, or a certain percentage have brown hair, or a certain percentage have green eyes, things like that. So we may have traits in common, but yet all of us are different, but together we make up the whole population. Now, sometimes I get asked, well, what about, aren't there hybrid alfalfa varieties? What again here? No, my what I'm going to tell you today. A lot of this, some of this is my opinion. So sort of disclaimer. My opinion is that in one sense, you can think of every seed in that bag is to some degree a hybrid. It has one male parent and a female parent that come together, and there's just really low levels of hybrid vigor in that seed. Just like you could say each one of us is actually a hybrid from our parents, but we're just exhibiting maybe a very low level of hybrid vigor, and that's why we're not giants walking around. So there are breeding strategies that some companies use where you can increase that amount of hybrid vigor you get a little bit. I guess I'm just not one of them that has used that, and I haven't seen enough benefit to really pursue that, but some people do. One of the things I wanted to mention was that, just like no two farmers do everything the same way, I guess no two alfalfa breeders do things exactly the same way either. Everybody has their own opinions about what's best. Not that one's right and one's wrong. Just varying degrees and shades and things like that. So with alfalfa, we're characterizing it by percentages and percentages for different alfalfa pests. So we run standardized tests for different diseases and insects. We find out the percent resistant of plants, the number of seeds that are resistant. So in a bag of seed, to call it resistant for something, say like Phytophthora, we have to have 31% of those seeds that are resistant. Now you say, well, that doesn't sound like a lot, and maybe it's not. But when we have to do that for a lot of diseases, and you also have to remember how many seeds per square foot. If you're planting 100 seeds per square foot, by the time we get to that end of that second or third year, we're down to 10, 20, maybe even less number of plants. So that's where we lose a lot of plants. We start off with a high number of seeds. To be called highly resistant, we have to have at least half of that bag, half the seeds in that bag have to be resistant. So we classify them with persistence or a percent resistance. We convert that to a resistance class. And then we're going to assign a disease resistance index to that. So we look at the 7 major diseases. We've got 3 wilts, some root rots, 2 races of Phenomyces, and anthracnose leaf disease. We convert each of those classes. We assign a number of points, 5 points for HR, 4 for R. So our maximum DRI is 35 out of 35 if everything is in HR for those 7. So do you really need a 35 out of 35 all the time? I guess I would say not necessarily. I like having at least an R for everything. And then maybe an HR for the things that are more prevalent in my area of the country or on my farm, things I've seen before or think I might have. But it's not critical to have a 35 out of the 35. So we're breeding better alfalfa. What do we mean by better? Well, we all want higher yield that in anything, I think, any crop. We want disease resistance. We'd like to have that 35. We'd probably like to have some insect resistance so that maybe we don't have to spray if we have at lower levels. We want it to be more nutritious, whether that's higher RFQ or NDFD or crude protein, whatever you like, but we want it to be more nutritious. We need it to be palatable. So the palatability maybe comes more into play when, depending on how you harvest and preserve and store that and the environmental conditions going into that. But if it's not palatable, then the cows aren't going to eat it. And we also need it to be persistent. We want it to survive 3, 4, or more years. So what are the significant pests of alfalfa? Well, for insects, there's a lot of different types of aphids in different areas of the country. At lower levels, there may be just more a nuisance than a real yield robber. Potato leafhoppers start in the Mid-South every year. They ride the air currents up the Mississippi, and then they head to the Northeast where they see those every year. Alfalfa weevil, some years it's bad, a lot of years it's not. Certain areas seem to get it more than others. For nematodes, the biggest one is the stem nematode, which right now is confined to the western states, west of the Rockies. We can kind of think of that. It's similar to the soybean cyst nematode, where you don't really know you have a problem. You don't know you have it until you see a problem. But we're trying to incorporate resistance in that and stay ahead of that. And then for diseases, the ones we mentioned, where I am in Wisconsin and in the upper Midwest here, Minnesota, phenomyces is probably one of the more prevalent ones. And even though it's a root rot, a seedling disease, a lot of times I've seen it even show up residually in that 2nd and 3rd year where it can impact your yield. And this picture on the right is one of the newer strains of anthracnose that we've seen the last couple of years. Shows up more towards the late summer. Even varieties that we have that are resistant to anthracnose, we've seen this in. So when we challenge the plants as breeders, we can do a lot of the disease and insect work in the greenhouse, and that works really nice. In the field is where we need to do the testing and screening for things like yield, the dormancy, winter survival, and quality characteristics. So when we're breeding, we have 3 basic types of breeding procedures that we're going to use that I'm going to talk about. The first is this phenotypic recurrent selection, which is population improvement. And so we're using mass selections where we have at least 80 parents, and we can have upwards of 200 total. And we're trying to shift that population for a given trait or traits without losing the other traits. So we want to move forward in one trait without going backwards in the others. We want to hold court there. We can also use genotypic selection, which is the progeny testing way to go, where we make what we call synthetic varieties out of a much smaller number of parents, but we know more about each parent and the genetics of it. And then finally, we got some techniques in biotechnology that we're incorporating into our breeding procedures. So the basic breeding strategy is just a big cycle where we start by selecting plants that we think are good plants. We cross them, we harvest the seed from those plants, inoculate it with various diseases and things. We take that seed and we plant it out in the field, evaluate it for yield, and the other diseases. And then through that evaluation, we can tell if we've made any progress or not. And if we've made enough progress, we may go and start increasing the seed there so we can tell the farmer. If we're still lacking in one or more traits, then we run it through the cycle again. And so we've always got something at some stage of this cycle or in the pipeline. So with phenotypic recurrent selection, it's the simplest of all methods, which is one of the reasons I like it, I guess. It's a quick way to respond to new pest pressures like that new anthracnose that we've seen. A relatively short cycle time where we're doing a lot of this work in the greenhouse over the course of a few months. It's relatively cheap to produce. I can guarantee you that all the breeding programs use this to some extent. The limitations are using it to increase yield, quality, winter hardiness, the field traits, things like that. And this process, it doesn't really look at the genotype or the genetic load, the genetics of the plant. It looks at the phenotype, which is what the plant looks like. So we want to pick good plants that look good. We don't really care why they look good, I guess you would say, or the genetics that they have. We assume they have good genetics if they look good. We're selecting them based on their looks. So the way we do that is I'm going to start out in the greenhouse and I'm going to take one variety and let's say I'm going to plant 10 trays with 1,000 seeds each. So that's 10,000 seeds total. And I'm going to hit those plants in the seedling stage. I'm going to start putting anthracnose, Phanomyces, Phytophthora on them. I'm going to put some aphids on them. I'm going to hit them with the wilts. I'm going to try and kill all the susceptible plants that I can. What I want to do over the course of a couple months is get down to the best 1,000 plants that survive all those diseases and insects. So I'm going to take those 1,000 plants out to the field and I'm going to put them in a nursery. So I'm going to plant them spaced out on coordinates so that I can identify each individual plant between each cut. And then each cut, I'm going to go and I'm going to evaluate that plant. I'm going to score it for how it looks. How is it regrowing? Is it leafy? Is it stemmy? What's the color? Does it have leaf diseases? Is it standing up? Did it fall down? Any number of different characteristics. So over the course of 2 or the next 3 years, I'm going to get that when we go from 1,000 plants, I want to pick out what I think are the best 300 plants. And here again, every breeder is different. So you end up with an Excel sheet. I've got range row coordinate for each plant, so I know where it is out in the field. And I've got a number of scores that I've scored it for. So somehow I have to look at this and pick out the best 300. And another breeder might have a different idea of what's the best than me, but that's all right too. I'm going to pick out what I think are the best 300. And then I'm going to go out in probably one of the cuts where it's getting to first flower, and I'm going to sample those 300 plants for quality so I can see what their quality is. I'll go out and I'll cut the entire plant off, put it in a paper bag, dry it down, and then grind it, and then run it through my NIR. So through the NIR, and I'll get a report that looks something like this, where I've got different sample numbers representing each different plant in the field. And then again, because we're running our own, I can run any number of traits. I don't have to worry about limiting that. Some of these I'm not going to use or aren't they're really that useful to me. I mean, ash depends on how much dirt is in it. It's not really affected by the genetics. Some of these are calculated traits. And again, different breeders will look at different traits with different weights. So what I want to do is there's a lot of numbers here, so I got to kind of figure out what do I want to look at, how do I want to look at it, so that I can pick out the best plants from these. What I like to do is I need a range of plants. I don't particularly care whether the range is from 200 to 240 or 120 to 160. When I'm selecting plants as a breeder, I need a range of plants so I can pick out the best from that group, whatever that group is. And so I'm going to convert, what I like to do is I make my own Excel sheet and I convert these numbers. I find the average of the 300 and I kind of standardize that so that I can sort by different traits. I can put a color code to it, and I can say, okay, these top ones, are above average for RFQ. They're lower for lignin, they're lower for undigestibility, they're higher in crude protein. And then I can go and I start checking off which plants I like so I can pick out my best 100. So remember, we started with 10,000 seeds, we got down to 1,000 plants, then we got down to 300. Now we're down to the best 100. So I've got a list of plants. I know the range plot coordinates. I can go out to the field. And about 2 weeks after I cut those plants off, they start regrowing. I'm going to go out and I'm going to take stem cuttings. Now I could dig up the whole plant and bring it in and try and cross that. But what I like to do is go out about 2 weeks after I cut back, and I'm going to cut and take the top 3 to 4 inches of stem from that plant. And I'm going to bring that into the greenhouse, and I can stick that into some potting soil in a tray and keep it moist. And then on the right, about 6 to 8 weeks later, I've got a nice plug with a nice root ball on it that I can pot up and use for my crossing. So on the left here, you see I've done that with a number of different trays. These are from a number of different varieties in the field. And I've taken multiple stems from the same plant. So I've got 6 genetically identical copies or clones of that one plant in the field. Now I've got those in the greenhouse. And I'll take one of those and from each of those 100 plants, put it in a pot, let it grow up and flower, and then I can start crossing it. Now, by taking multiple copies in the field, what I can do then is I can start, for instance, I could take the top out of those 100, I could take the top 20 for RFQ from this group of 100, and then I could take the top 20 from 4 other varieties and combine those, and I can start to make different combinations and different new experimental varieties that way. So once I've got these in pots and up and flowering, I can take them into my growth room inside, and this is typically in November. So when it's cold outside, we can do crossing inside. I can bring those plants into the growth room, and in there I'll have a hive of bumblebees. And those bees, by controlling the lights, I can get them to come out and interpollinate these plants for me. And then I can turn the lights off, they'll go back in their hive. I can move the plants out and bring in a different cross the next day. They'll go into the hive, clean the pollen off their legs, and then come back out the next day ready to pollinate again. So the bees do a lot of work for us. They're a big help, and I can say in out of the 20, 25 years I've been doing it so far, I've not gotten stung by a bumblebee. So once they do their pollination, we take the pots back out into the greenhouse and we let that seed mature. It starts to form the green seed curls. And then when they turn brown, we can go in, clip them off, harvest them, thresh them, and take that seed. That's the seed then that we use in our yield trials and for further disease screening. So how long does it take to go through that whole process? Well, if you've been keeping track from when we started screening in the greenhouse, selecting our plants, making our cross. After we make that cross, then if we're going to yield test it, we need to test it for at least 3 years in the field. And then we're going to start, if we're going to pick from the experimentals we have in our yield trial, we're going to pick the best one. We're going to start increasing our seed, produce foundation seed, our parent seed. And then the next year from that, we can produce certified seed to start selling to farmers. So you add that all up and we're probably at 8 to 10 years a lot of times. So it's a slow process, which is part of the reason it can be fairly costly. Now, can we speed that up at all? Well, we can, and we can we have at times using a model similar to Operation Warp Speed that they've been using for the COVID vaccines, where they're producing the vaccine at the same time they're doing the clinical trials testing it. So we can do the same thing. We can produce our foundation and certified seed while our variety is still in the field being tested. The problem is there's risk involved in doing that because you could get something that doesn't pan out, and then you've wasted a lot of money producing your certified seed that you're not going to be able to sell. Certain times there's market conditions that would warrant doing this. or if you feel really good about a certain variety, you can take a chance and go through this. But this is more atypical than typical, I would say. So here's an example of one of our field yield trials. This was taken from one of our Pioneer plots when I was with Pioneer. This is actually happens to be one of our leafhopper resistant trials. So you can see the dark green are resistant to the leafhopper, and you see the yellowing in the other plots. So our yield trials, we plant plots 3 feet wide, 12 feet long, 4 replications of the same variety randomized throughout this block. We'll seed this at 8 locations and for 2 consecutive years. And then we want to get at least 3 harvest or production years from those seeding years of data before we can move on. So the genotypic selection process or model, we can think of similar to dairy and beef, where we do bull testing for artificial insemination. Only in alfalfa, instead of the bull, our alfalfa plant is really the mother plant, the female side of it that we're looking at. So we intercross a bunch of mother plants together. So they're bred to many different mates or random pollen donors. So in theory, they should all be about the same. And then we're going to harvest seed from those individual mother plants to see how the progeny do or how the offspring look, either for disease or in yield trials. And then we want to select the best mother plants that we can find that have the best progeny. And then we're going to intercross those. The only difference now with alfalfa versus maybe dairy is that with alfalfa, the plant is both the male and the female. So we're harvesting seed off it as a female. Remember that plant we harvest seed off of also contributed pollen to all the other females. So if we take a look at an example of this, you see this, each of these kind of rows represents seeds from one of the mother plants. So this one on the left that has good growth, nice tall green growth, I think this may be like a flat that had Phytophthora in it. So we're going to look at that and we'll say this mother plant on the left has genes for Phytophthora resistance. The one in the middle that looks short and stunted probably has none. So we don't want that one. And then over to the right, we've got rows that they've got a few good plants, but not too many. So maybe they had some good pollen. You know, those good plants came from pollen from this good mother. We just don't know. But what we really want to do, we could make progress for one disease doing it this way, but we would really need to test for all the diseases and then find plants that are high for all the diseases. But instead of doing that, what we try and do is start with plants that have been through our cycling for disease that we know have high levels of resistance. And we're going to use this process to try and increase our yield. So instead of this being a flat of Phytophthora, think of this as a yield trial, where this one on the left would be your growth in the field. So we want to find mother plants that look like that in the field that really look good for yield and combine those. So we want to find those good mothers, intercross those together, and then hopefully what we should have is a variety that's a lot higher yielding than what we started with. We still need to test that variety for yield for another 3 years then. At the same time, we're going to test it for the diseases to see if we've lost any of the diseases. If we have, then we'll need to put it through that cycle again, bring it back up so that it's on par with what we need. So for the genotypic, the advantages are it's the best way to improve things like yield and persistence, where there's a lot of different genes at work there, not just one. And it also works well with biotech because we can deal with a lot lower number of plants, but we know more about it. The disadvantages is that it takes a long time to do that because it takes 3 years to see which mother plants are the highest yielding, and then another 3 years to evaluate it to prove that we've done that. And that makes it very expensive. So I just wanted to check or review quickly. You've probably all seen this yield versus quality graph, where our forage yield is the red line. Forage yield made up of the stems and the leaves. The stems is typically about 2/3 of our yield. The leaves have more of the quality. We know as the stems get more mature, they decrease in quality. Um, as that plant matures, it's going to go from a vegetative state to reproductive, put its energy into reproducing. It's not going to grow anymore. At some point, it's going to get big enough. Those lower leaves are going to turn yellow. They're going to start to fall off. We'll lose yield and quality there. So, what we're really trying to do is find that spot where we can get the most yield, but not lose quality. And in developing varieties to do that, if we typically start with a variety, say that is this red line, what we're trying to do is shift that line higher. So we start at a higher level of quality to begin with, or we start at the same level of quality and then just have it decline at a slower rate. So we're trying to do one of those two things. I also just wanted to put a quick plug in here for if you're still looking at RFV, I'd encourage you to switch and start using RFQ. There's a good article in Hay and Forage Grower magazine or an editorial. There's a push in the industry to get away from even really looking at ADF and going to the RFQ, where we look at the individual or the digestibility of the individual components that make up our plant. So I encourage you to try and use that as much as you can. So next, I want to talk a little bit about new technologies that we're using, and there's really two areas. One is, this genetic modification by insertion, inserting new genes either from a different plant species or maybe from a bacterium, as we've done. And this is what is known as, the traditional GMO, where in alfalfa, so we've used it. We have the Roundup Ready gene that has done this. They started with soybeans and then went to corn, and eventually got around to doing it in alfalfa and some other crops. The last couple of years, we have the Harv Extra reduced lignin gene, or reduced lignin trait that's been out. And then in the future, there could be GMO traits more coming out. I'm not aware of any at the moment that are imminent, but there could be. So we've got inserting GMOs, and then the other way we do it now, more recently, is we have this gene editing technique, which is going to be used in a lot, I think, in agriculture, also the medical and therapeutic fields. They're looking at a lot of different uses for that. And there's two platforms for that. One that you've probably heard of is the CRISPR. It's gotten more media attention. And then also the TALEN technology, which is similar, but it's a different way of doing the gene editing. So what is gene editing? It's deleting or replacing part of the DNA at a specific site in the cell. And to do that, they use two types of nucleases, which is what this CRISPR and TALENs are, that I refer to as genetic scissors. So the CRISPR-Cas9 that you've heard of, it's simpler, it's cheaper, it's not as precise as the TALENs, and so we tend to get more off-target effects. So we have to go in and check and see if there's something has happened that we didn't intend, and then if there is in that plant, we have to throw that plant away. As far as I know, Pioneer and now Corteva owns a large number of the agricultural patents using this CRISPR-Cas9 technology. What's held it back really is there's been this battle between this group on the East Coast and the group on the West Coast over who really invented it and invented it first and has the rights to it. So that's really slowed down. I know they've distributed this so a lot of people can do it and use research, but there really haven't been products out there with that technology yet. The TALENs is another way of doing it. It was invented by a guy that started this Calyx company out of Minneapolis, Minnesota, and it's now owned by the Selectus Group out of Europe. Excuse me, but Calyx has the license for the agricultural products with Selectus, and by teaming up with them, we have access to that. This TALENs technology, I call it ultra precise compared to the CRISPR-Cas9. And the way I wanted to kind of explain, the way I thought of that was the CRISPR-Cas9 would kind of be like, if you remember, like in kindergarten, if you would take that small safety scissors with the dull points on it and try and cut construction paper. That's kind of what that's like. Versus the talons is more like that sharp scissors that your mom kept in that special drawer that she told you not to mess with because you were going to cut yourself. Really now the CRISPR-Cas9, there's been a lot of research done on that. It's getting better. It's actually more like maybe the scissors that's in your toolbox that you use to cut twine or something like that now. But there's a lot of research in both of these areas. They're getting better as we go. So I just want to take a look now. When we're thinking about this, we're looking at a plant cell here. And so we've got a plant cell. The outside of the cell is made up of the lignin, which holds it together, gives it structure, and that holds the good stuff in the middle that we want to get to, that our cows can use, that they want to get to when they eat. We've also got a nucleus with a chromosome, and the chromosome has genes that tell this cell what it should be. Is it a root? Is it a stem, a leaf, a flower, whatever? So if we could go in and take that chromosome and we grab a piece of it and start unraveling it, it's all tightly wound and wound and wound, and we unwind it. Eventually, we're going to get miles and miles and miles of this double strand of DNA. So there's two strands, and they're connected by these letters A and T every so often. So one of the ways a colleague of mine came up with this, but one way to think about this is if you think of a chromosome like an interstate highway, like I-35 going from Duluth down to Laredo. And then we have different genes on that chromosome. And those genes are like the different cities along the way on that interstate. And so through like a lot of university research, we know where some of those genes are. So we kind of have a map, and we know what some of them do. Not all of them, but we're getting better. So just like an interstate, you've got a northbound lane and a southbound lane. With the DNA, we've got two strands. We've got one going northbound, and we've got one going southbound. And then they're interconnected in the middle at all these different spots. So where they're interconnected, we can kind of think of that like those crossovers in the interstate where the state patrol sits and watches for speeders. So if you've ever been driving down and you see, and there's two of them sitting there, you'll have one in a red car who's watching the southbound lane, and one in the red car or a blue car that's watching the northbound lane. And they're kind of in a pair. So this kind of represents our base pair of nucleic acids. So we've always got an A car or a red car matched up with a T car, or we've got a red G car matched up with a blue C car. So they're always sitting in together in a pair in the middle. That way, they can kind of talk through the window to each other. They don't have to communicate on the radio and let us know that they're sitting there. So what does that mean when we're doing things? So each of those combinations makes up a base pair, and they're all along those crossovers. And if you're driving from north to south, you're going to go by and you're going to see, and there's going to be a certain sequence of A's and C's or whatever. And what we're looking for is a certain sequence that tells us something is coming up or something's going to happen. There's a sequence that codes for something. Now in alfalfa, there's along that interstate from Laredo up to Duluth, there's 800 to 900 million base pairs or spots where we have those squad cars sitting. So that's a lot, and a lot of them really don't do anything. They're just kind of decoys. We have a lot of that junk DNA that just kind of takes up space. But what we're trying to do is find and look for a certain sequence, a certain combination of letters that tells us something. So in alfalfa, what we're looking for is something that codes in this lignin pathway. Now, the universities have done a good job of documenting this lignin pathway. They know that there's a certain gene that codes for an enzyme that is used by compound A to convert to compound B, and then that goes to compound, uses another enzyme and gets converted to lignin. So again, if we think of these different genes on that chromosome, on that highway, like different cities. So for instance, Minneapolis might be this gene that we need to codes for an enzyme. Well, if we use the GMO insertion technique, we're driving along that highway and we see the exit sign for Minneapolis, but we've had some vandals and they've come in and they've inserted some extra letters into that name. And now it says Minneapolis. So we're driving along and we're like, well, that's not Minneapolis. So we keep on driving. We bypass Minneapolis. So this gene gets bypassed. It doesn't get turned on. So we don't get this enzyme one that we need to convert the compounds, and we end up with reduced lignin. Now we can do the same thing on the right by gene editing. We can come along and have somebody take out some letters from that Minneapolis. And now it says Mippolis. Well, Mippolis is not Minneapolis, so we keep on driving. We don't stop at Minneapolis. The gene doesn't get turned on. It doesn't make the enzyme, and therefore we don't have as much lignin. Now, all of the cars are not going to go by. Some will figure it out and say, okay, this is Minneapolis. We need to turn this gene on. So we don't want to get rid of all the lignin, because then we wouldn't have any structure to our plant. We'd have nothing to hold it together. But we want to reduce the level of the lignin in that plant. So what do we know about lignin? Well, we know that the lignin content in the plant cells increases with the maturity of the plant. We know that the lignin is indigestible, and that's bad for us anyway, for the cows. And it's going to bind with the cellulose and hemicellulose inside when we chew it up, thereby reducing the overall fiber digestibility. So what can we do about that? Well, if we improve the fiber digestibility and quality, we can improve the fiber digestibility and quality by reducing that lignin content. And we do that through these biotechnology ways by either not what we call knocking out or silencing or inactivating the genes in that lignin pathway. So what we've done, S&W together with this Calyx Corporation, is we've provided the alfalfa plants to them. They go in and they do their gene editing magic, and exactly how they get that nuclease to go in and cut at a certain spot in there, I have no idea. And I'm sure there's molecular biologists and stuff that would cringe at my explanation of this, but hopefully it makes some sense to you. And then so they'll go in, they'll work their magic, and they return to us then a plant that we can use and start breeding with. And with gene editing, these are going to be non-regulated, non-GMO plants. So why do we want to commercialize that? So, initial tests on our populations have shown that if we get up to a 10% reduction in whole plant lignin, we can get a corresponding increase in overall digestibility. We need to do that by still holding our yield and pest resistance comparable to our standard varieties. But if we can do that, then we can get increased milk and beef production out of the same amount of feed. And also we can get decreased manure production, which would benefit us. I know here in Wisconsin, one of the big things is the large dairies have a hard time getting rid of all their manure. So again, by reducing the lignin, or I reduced lignin alfalfa, if it had equal or greater fiber digestibility than alfalfa harvested 7 to 10 days earlier, what benefits could we see? And there's really two areas. So we can either increase the likelihood of harvesting alfalfa with higher forage quality that we can either sell or use ourselves for our animals, or we can improve the flexibility of our harvest management while still having the same quality, but possibly higher yields. Or if we can reduce our cuts, if we could go from a 5-cut to a 4-cut, or 4-cut to a 3-cut, what are your costs per acre of a harvest? I know here in Wisconsin, especially if you do custom harvesting, it can be anywhere from $50 to $100 an acre, I believe. So the advantages of this gene editing technology over the GMO insertion technology, again, these gene edited traits right now and are likely to be non-regulated, so that they're cleared for most countries. And that's a big deal in the West, where states like Washington, California, Arizona, they export a lot of hay to the Asian market. There's reduced cost for development and a faster development time, and that's largely due to not having to do the regulatory studies required by the FDA, EPA, or USDA, because those cost a lot of time and it costs a lot of money. And really, the reason we can do that is because what we're doing is modifying genes that are already in the plant. We're not introducing new genes or new or foreign DNA. Working with what the plant already has, essentially we're kind of looking for that mutation, like 1 in a million mutation that a plant would naturally have. We're just kind of making it ourselves in a spot where we know to look for it. And then we can use that in our breeding program. So right now, we're in the early breeding stages yet for fiber digestibility using this technology. Our preliminary data showing we're on target to improve quality in a wide range of dormancies. The first products coming out with this improved quality in the gene editing are probably going to be in that 7 to 8 dormancy range. We're in the towards the more non-dormant side of things because there hasn't been a lot of work done in improving quality on those varieties as there has in the dormant varieties. It's non-GMO, which is going to hopefully should extend our market acceptance around the globe to many different countries. And this really represents the first forage that contains a gene-edited trait. The Calyx company, they've used this technology. They started in potatoes. They've used it in soybeans for oleic acid, I believe in canola. And so now they're trying to help us with the alfalfa part of it. I just want to finish up. If you want more information about S&W, you can go to our website here. Alfalfa Partners is our alfalfa sales and marketing division. Ward Labs has a lot of good information on their website. If you want to learn more about the consortium and what they do, you can go there. hayandforage.com is Hay and Forage Grower Magazine. A lot of good articles in there. Progressive Forage puts out Progressive Forage Magazine. And I know Becca has a nice article in there this month too that you should all take a look at. So I guess thank you for listening to me for a while here. And I hope some of it made sense, probably not all of it, but gives you an insight as to what's going on in the alfalfa breeding world these days. So any questions, Becca? I'm afraid to ask.
Yeah, so first of all, did you have a slide with your information, or do you want me to just try and, forward people who are looking to get in contact with you?
I didn't make one with that, no. So
okay, so we do have someone who is requesting the PowerPoint file, so I can get with you later on that to address that. But first, let's start with our first question that is actually in the Q&A. And I think it's a good, after we've just had all that good technical knowledge, to do a good refresher on this. So the question is Harv Extra gene a true GMO? For organic producers, they aren't allowed to use GMOs, but they think CRISPR is allowed, but it's not considered GMO. So first of all, looking for that clarification on the Harv Extra and then on the CRISPR. And this was asked before you really went into that. So I think just another clarification on that would be helpful.
Okay. Do you want me to stop sharing?
Oh no, you're good.
Is it better?
Okay.
Otherwise you can see me. I would say yes, the Harv Extra is definitely a GMO trait. So if you're organic, you do not want to be that. The other reason is the Harv Extra trait is tied together, linked with the Roundup Ready trait. So you cannot get Harv Extra without Roundup Ready. So if you're organic, that's probably a big no-no. And right now there are no products with the CRISPR technology for sale that I know of. I would anticipate when it comes out, it will be the same as with the Talents technology, that it will be non-GMO. I anticipate that, but I don't know that for sure. So let's see.
Okay, let's move to, we got a question in email before this started, basically to summarize for our viewers. The producer has a lot of land that already has existing alfalfa growing, very much thriving on their land. They've tried other crops unsuccessfully. They're looking to figure out how do we go about identifying varieties that are already existing and already doing well in their environment so they can plant more.
Yeah, and I guess identifying varieties in the field is very, very hard. So unless you know what was planted, it's probably an exercise in futility. And you can get certain clues from flower color, but a lot of it is going to look the same in trying to figure out the traits. What I would say is that even if it's grown well before, I would suggest looking at new varieties, because we're constantly making improvements, and new varieties are going to be good. If other crops don't grow good, I guess I would look at some of the agronomic things, or the I would start by doing the soil testing, maybe contact your county agent or an extension specialist to have them come look at it, to get some more localized help to see what's really going on there, why things aren't growing. Before you plant, you want to make sure you have the proper pH. That's one of the most important things, and then have a proper level of potassium, of K, to get your crop off to a good start. Sulfur is probably the other nutrient that we've seen in recent years become limiting, in established stands. But starting off with, you need a good seed bed, have it at the proper pH, and have some soil nutrition in there to start with to get it off to a good start. And then using I would say good varieties. I know you can still buy Vernal and Ranger and things like that, but I would advise against using things like that. I would go for commercially more recently released varieties that do have seed coating on, for at least for fungicide, and most of them will today.
Great, thank you for that answer. Okay, so then from Chad Hansen, and this is a NIR related question. If wet chemistry is the gold standard, the most accurate results, why not use wet chemistry lab analysis instead of NIR for plant breeders? So I'll let you address that specifically as it pertains to plant breeders, but I did also refer him to the consortium did put out a webinar presented by Dr. Uttam Saha out of the University of Georgia. He really did put together the data and the case for why NIR can be advantageous in certain scenarios, but I'll let you go ahead, David, as to why specifically for a plant breeder this is advantageous.
Well, I guess again, the short answer would be time and money. Wet chemistry can be very expensive. The chemicals and reagents, and the time doing that, and then running a separate wet chemistry analysis for each constituent that you want. Where with the NIR, you can scan in and get a lot of them just by doing one scan. And a lot of times what I'm doing is I'm taking a sample on right before cut 2, my second growth. I'm going to sample that and run it through the NIR, and I want results by the time I get to cut 3, because when I cut it back shortly after that, that's when I want to take my cuttings and bring it into the greenhouse. So and we're doing maybe we're doing 300 samples for that one variety, but we're probably doing several thousand samples overall. So using wet chemistry is probably not realistic. And again, I would argue that using NIR is just as, and again, and I know in like you said, Uttam's presentation, he talks about, I think, accuracy versus precision. And if I have this right, when I do stuff, I don't need to be accurate. I don't care if it's 140 or 180. What I want is to make sure that with the samples that I run, they're all the same relative to each other. So if I'm shooting, they're all kind of hitting the target in the same area. Maybe it's not in the bullseye, but they're have a small pattern. So I'm looking for that comparison between the varieties, not the absolute value. Versus compared to maybe your lab, you're more concerned with the accuracy of that. But I think with NIR, we're doing a pretty good job of getting both of those now.
Yeah, absolutely. And like I said, watch Utam's presentation and you'll get a really good idea for how far that technology has come and how reliable it is as a tool for analysis. And then of course, later in this webinar series, I'll be presenting and talking a little bit about why NIR is acceptable. Okay.
Becca is much more knowledgeable about that than I am. So
you kind of touched on this a little bit, the dilemma, yield versus quality. So we did have an email comment question coming in asking about if you had any suggestions as for cutting height, and they were wondering straight alfalfa as well as alfalfa grass mixes, and then also wondering about moisture. But I think we'll get into some of that when we do our producer panel. But if you had any thoughts on those kind of production concerns, you can go ahead.
Sure. Well, and I think, a lot of it too, I mean, you can take a cue from your nutritionist, what you need in your ration. Do you need higher quality? What level of quality do you need? Are you, and what is it for? Is it for high producing dairy cows, for beef cows, for heifers, something different like that? I guess what I say is if it was me, I mean, I like to plant the highest yielding variety that I can that has maybe adequate or average quality. As much as we try and do with genetics, quality is still a big part of it is still management. It's still the cutting schedule and how quickly you can get that forage put up and processed at a high level before it starts to degrade. The better you can do that, the better feed you're going to have. So if genetics is not going to make up for bad management. That's really where you have to you can focus. Once you do that, or if you're having trouble there, you can go for those high yielding or high quality varieties if that's what you really need for your dairy. But it's hard to compensate to make up for yield if you don't have that to start with. And again, everybody, every situation is a little bit different. That's why there's no one size fits all for everybody. That's why you see tons and tons of alfalfa varieties. A lot of them claim to be the best, but they've all got, pluses and minuses to them somewhere.
Absolutely.
So hopefully that kind of answers the question.
Yeah,
management's a big part of it.
I don't think it's a question that you can say, straight 6 inches that's the best. It really depends on the situation and the genetics and the management.
The cutting height, it depends on your geography. And again, how intensively are you cutting that? Are you taking 5 cuts? The closer you cut it, maybe you have a little bit slower regrowth. How quickly are you getting that forage off? Are you getting it off before it starts to regrow? And a lot of that depends on the equipment you have to cut too.
Yep, absolutely.
But I don't know. For me, a good rule of thumb, is that 3-inch range. I like 3 inches. But there's a lot of variability can be in that.
Definitely. Well, thank you once again, David, for presenting some really cutting edge technical information there. I really enjoyed it. Hopefully the producers and forage, utilizers that have gotten on have also. I'm going to go ahead and plug us here. We will be hosting a month from today, Monday, again at 1:00. And we're going to be having Rich Russell from Snw Seed. He will be presenting, and he's going to talk all about, choosing the right forages and choosing the seed for your operation. And so I think that'll be a really great next step for what we just learned about how the seeds are produced and how they're bred and all of that great information that we got from you, David. And then we also have another webinar coming up actually later this week. One of our support agronomists, Casey France, is going to be presenting at High Plains Journal's Alfalfa U. That's going to be on Thursday, the 18th. She's going to talk about how alfalfa might actually be a solution for some marginalized soils. And there's going to be some other, really great speakers, Dan Undersander from University of Wisconsin up in your neck of the woods there, David. So with that, I think that concludes today. Once again, thank you so much. We really appreciate, the time you put into that presentation. And I think that's all for today. So thank you.
You're welcome. Glad to be here.